Thank you so much for joining us for today's edition of Lunch a la Carte, where we get to hear from one of the CART Fund researchers directly from them with information on the status of this disease and their research and things that are going on around the country. And we are so grateful that each and every one of you have taken the time to join us today. I just wanted to give you a couple of quick announcements to remind you that the end of the fiscal year is only 5 days away. And as of today, we sit at $1.936 million raised for this year. I know there are some major gifts that are coming in this week. I have no doubt we will pass 1.95, but with your help, we can get even higher than that. We've already exceeded last year's donations, and we are so grateful for everything that you guys do to help us. Did want to give you some quick announcements. The next edition of Lunch a la Carte will be on July the 17th, which is a Wednesday, and that is a noon Eastern Standard Time, as always. So we hope that you will put that on your calendar, that you will share it with other people in your clubs and in your districts, and even on your social media accounts to invite people who are non-Rotarians as well. Beginning in July, we will start a new schedule for our monthly CART Conversations calls. Those will be held on the third Thursday of every month. So go ahead and mark all of those dates on your calendar as well. Again, 5:30 to 6:30 p.m., which is our normal evening meeting schedule. We have some great speakers and topics lined up for that moving forward this year. Again, that will be the third Thursday of every month, and those dates are already on the calendar. You can find them in DACDB under multi-district events, or you can find them on the CART Fund website under our calendar. If you haven't looked at the website lately, I encourage you to do that. We're making changes daily. We've got some new additions to it, including some new photos, some other new things, and some new merchandise on the CART page as well. We are also going to be hosting our annual training session for all CART volunteers, no matter what your role is. If you are in the district governor line of your district, if you're a club district chair, if you are a club CART chair or a district CART chair, or even if you're just someone in another Rotary Club who is very supportive and interested in learning more about CART, we would love to have you join us for that. That is going to be on Monday, July the 29th at our normal meeting time of 5:30 p.m. That is a Monday, July 29th from 5:30 to 6:30. We encourage you to participate in that. We will record it and share it to YouTube afterwards, but being on with us live gives you an opportunity to ask questions and make sure that we haven't missed anything. We'll be rolling out some new branding, some new merchandising, some new fundraising opportunities, and most importantly, we'll be sharing with you some new PowerPoint templates and things that you can use as you promote CART this coming year as well. So we are very excited about all that's going on. And at this time, I want to ask our immediate past president for another 5 days, our own Bill Parker, if he will introduce our guest speaker for the day.
Thank you very much, everyone, and thank you for coming. Dr. Vogel comes to us from sunny California. When he got his CART award, he was in Minnesota, and I guess he saw the light and went to warmer climes. Dr. Vogel is the Michael M. Minchin Jr. president. He's the J.D. French Alzheimer's Foundation endowed chair. He's the director of the Alzheimer's Disease Research Center Biomarker Study and professor of neurology at UCLA. Dr. Vossel received a medical degree from the University of Tennessee in Memphis. He completed his neurology residency at Harvard Medical School and Massachusetts General Hospital and Brigham and Women's Hospital, and fellowship training in behavioral neurology and dementia research at the University of California, San Francisco, and the Gladstone Institute. Dr. Vossel investigates Alzheimer's disease with a focus on brain rhythm abnormalities and translational therapies. Dr. Vossel has contributed articles to the New York Times, the Los Angeles Times, and has been featured in numerous national and international media outlets, including CNN, NPR, The Washington Post, the Boston Globe, Daily Mail, Canadian Broadcasting Corporation, Fox Local Channel 11 in LA, KNX News Radio in LA, Dr. Radio on Sirius XM Radio, and Spectrum News 1 SoCal. In addition to the CART Award, Dr. Vossel has received the John Douglas French Alzheimer's Distinguished Research Scholar Award, a part of the Cloud Transitional Research Award from the Alzheimer's Association, and the Bernese Epilepsy Award from the University of Bern in Switzerland, and the Outstanding Healthcare Innovator Award from the Los Angeles Business Journal. Without further ado, I would like to direct your attention to our researcher and CART Award winner, Dr. Keith Vossel. Sir, you're on.
All right. Well, yes, I was a recipient of the CART Award in 2018, and that was really a launching point for my career, and I had a wonderful time visiting you all down there. I'm actually from Tennessee, so it was also a homecoming of sorts for me. So it's really wonderful to have this chance to give you an update on the research that I've been doing in relation to seizure disorders in Alzheimer's disease and related dementias. I have no relevant disclosures. So as an overview, I will begin by giving you some background on Alzheimer's disease epidemiology and the pathophysiology. I'll go over the latest figures on seizures and epileptic activity in Alzheimer's disease. I will discuss some of the mechanisms that we have discovered in the laboratory for what causes these seizures in Alzheimer's disease, and then talk about the connection that we've recently discovered between late onset epilepsy, which is epilepsy that begins later in life, and the development of dementia. And finally, I will go over some of our current and future directions. So I'd like for you to learn a bit about the latest literature related to silent or subclinical epileptic activity in this disease, as well as recognize associations between epileptic activity and cognitive decline in dementias. Right. So we know currently that there are 6.7 million individuals in the US that are living with Alzheimer's disease. And this number is expected to increase at the current rate to reach 13 million by the year 2050. The current cost to our healthcare system is $345 billion. We're expecting this cost to increase up to $1 trillion by the year 2050. So we really need to continue to invest funding in Alzheimer's prevention and treatment, because even if we had a treatment that could delay the onset by 5 years, that would reduce these numbers in half. It's really a growing epidemic throughout the world, with 57 million individuals worldwide now living with Alzheimer's and related dementias. Rates are increasing exponentially in developing countries, even though the rates are starting to plateau in the US and in Europe. Another staggering figure is that up to 18% of older adults have mild cognitive impairment, illustrating that we really need to be more vigilant to detect cognitive symptoms in older adults. Not all of them are developing dementia, but this is eye-opening and shows us that we need to devote much more medical attention to our older adults, both in neurology and primary care. Over the last 100 years, we've learned a lot about this disease, starting with Alois Alzheimer's first patient over 100 years ago, when he described the clinical and behavioral symptoms of a young lady who was in her 50s, who began having memory and other issues. We've also learned about the regional volume loss in the brain, including loss of brain tissue in the cortex and the hippocampus, which is important for memory compared to a healthy brain, as well as the pathological causes, including amyloid beta, which builds up outside of neurons, and tau tangles, which builds up inside of cells. However, we haven't really discovered much about the neuronal activity changes until more recently. And this is vitally important because the neuronal activity and the way that our brain communicates across different regions is essentially what makes our brain click. and to know how that works might uncover ways that we can help the brain to become more resilient, even in the face of these pathological changes that are occurring. In the laboratory, we model Alzheimer's disease by expressing a protein. This is an example of what the protein looks like at the cell membrane. This is amyloid precursor protein, and it's generally sitting there in this outside of a cell, but it can also be cut into two pieces. There's one cleavage site up here and another down here. And after these two sites are cut, there's this part of this APP protein called amyloid beta, which is very sticky, and it gets secreted from the cell, and it can assemble into these aggregates outside of the cell and make clumps called amyloid plaques. Inside of these plaques, the neurons become entangled and and the cells nearby them begin to die. So more recently, we've been able to remove these plaques with new treatments called amyloid lowering therapies, and that is beginning to show some evidence of benefit over the long run. But there's much to do still, because it's only a modest benefit when you remove these plaques. So we need to continue to look for other treatment targets in the brain. So this is an animal model experiment with that mouse that's overexpressing amyloid precursor protein that ultimately leads to those plaques. And next to this mouse here is a normal mouse, or NTG. And so I'm going to play this video for you. And what I'm showing you here is a seizure that's occurring in this mouse model of Alzheimer's disease. You can see the electrical activity begin to build up, and then there's this chaotic looking brain activity that's quite high in voltage compared to the normal brainwave tracing. And while this chaotic brain activity is occurring, the mouse is frozen in its tracks. It's not even moving. So this is an example of a behavioral arrest seizure. And as soon as the seizure stops, this mouse wakes up and begins to move around again. And you can see that happening now. And while the mouse is moving around, you can see that the tracing shows very little brain activity compared to the normal mouse next to this mouse. And so what we're seeing here are bursts of seizure activity followed by prolonged suppressed activity. And we think that over time, this leads to impairments in the brain as it's trying to combat the seizures that leads to problems with learning and memory. We're also seeing evidence of seizures in another model. This is a model of dementia with Lewy bodies. And so this is a model where we're overexpressing another gene called alpha-synuclein with mutations that are linked to familial Parkinson's disease. And you can see that every time there's a spike here on the tracing, this mouse is having a jerk. This is called a myoclonic movement. And this is an example of myoclonic epilepsy, which we can see in Lewy body dementia. And we're able to suppress these epileptic spikes with an anti-seizure medication called vigabatrin. And we're testing that out now in our animal models with an eye towards future clinical trials. So when I began doing my research, even prior to the CART award, I was a fellow at UC San Francisco. And as a clinical fellow who was doing research in the lab, I was interested in whether patients might be experiencing similar seizures as our mouse models of Alzheimer's disease. And up to that point, we knew that up to 22% of Alzheimer's patients do develop seizures at some point in the course, with higher rates in younger patients and those with mutations that are linked to familial Alzheimer's disease. What we did not know is whether there could be more silent or subclinical epileptic activity occurring, for instance, during sleep, and whether this seizures could be happening in people in the earliest stages of dementia, because prior studies had only studied patients that were more advanced. So in order to do that, we looked at patients that came to our clinic, and this is a histogram of incidence of new seizures in patients as they're developing dementia. And you can see that the bars start to increase around time 0, and 0 refers to when people are beginning to have symptoms of dementia. We found that up to 13% of our Alzheimer's patients and 15% of our Lewy body patients were having seizures during the first few years of their disease. And the seizures tended to cluster around the beginning of their onset of cognitive decline, which was novel when we presented this and published this. So this raised awareness to start looking for seizures that could precede the diagnosis of dementia. And as you can see, there are a few cases of seizures that occurred up to 10 years prior to the onset of cognitive decline. The myoclonic movements that I showed you in the mouse also occurs in these different dementias at different rates, up to 60% in Lewy body dementia. And those symptoms begin a little later in the disease, about 4 to 5 years after the onset of cognitive symptoms. But this is another sign of the brain being overactive, because these movements can be suppressed with anti-seizure medications. So when we first looked at this about 10 years ago, the seizures that occurred in dementias were thought to mostly be motor-based, like the types of seizures you may be familiar with, like grand mal seizures, where there are convulsions and people lose consciousness. But what we discovered in our review of our patients is that the seizures that our patients with memory loss were experiencing were generally non-motor in nature, and they could have spells like deja vu, or just an absence type seizure where they're not speaking for a little while, or just other amnestic spells, or even confusion. And this could last for 30 seconds at a time. You may be familiar with the spells that Mitch McConnell had recently after a fall. It may have been due to that fall that he had, but he had these little spells that we think might have been seizures. Those are the exact kind of spells that we also see in these patients. And around 2/3 of the seizures that we saw were non-motor in nature. And they would be hard to detect if you weren't really looking carefully for them. One thing that was interesting about the seizures in these patients is that they associated with younger patients. And the patients that had mild cognitive impairment or Alzheimer's disease and seizures were 7 years younger on average than those without epilepsy. So this suggests that maybe the seizures are hastening the onset of the cognitive decline. So up to this point, we had seen that seizures of various sorts could occur in about 20% of our patients. But what we did not know is whether there could be additional epileptic activity like we were seeing in our animal models, especially at night when people are sleeping, in people with no known history of seizures. So to study that, we used some sophisticated detection technology at UC San Francisco, including a magnetoencephalography. It's like a helmet that people wear while they're resting, and it detects magnetic fields that are generated from electrical currents in the brain. And we also used EEG, which many of you may be more familiar with, which are these electrodes that are placed on the scalp and that can record the electrical activity directly. So combining these two methodologies gives us our best sensitivity to detect epileptic changes and other changes in the brain. So we used EEG overnight in our patients with Alzheimer's disease and age-matched controls, and we used MEG for an hour during the daytime. And we also were able to look at fluctuations and other cognitive and genetic outcomes in these patients. So we were actually very surprised to see that over 40% of our patients with Alzheimer's disease were experiencing epileptic activity during their sleep, whereas very few of our normal controls were experiencing epileptic activity during their sleep. There was a fourfold increase in epileptic activity in our Alzheimer's patients compared to the age-matched controls. And initially, when these people came to the clinic, those with Alzheimer's disease and epileptic activity looked similar to those with Alzheimer's disease without epileptic activity in regards to their cognitive testing. However, as we followed them over several years, this is in months, so over several years, we found that their scores on a test of global thinking abilities called the mini mental state exam declined much faster, suggesting that this epileptic activity is likely contributing to their accelerated cognitive decline. So this motivated us to think about how we could treat the epileptic activity and potentially improve the outcomes and slow the cognitive decline in these patients who are experiencing epileptic activity. Since our initial studies that were reported around 2016, there have been many groups that have followed up and found similar results around the world. There have been rates reported between 22 and 54% of epileptic activity in mild cognitive impairment and Alzheimer's disease, associations with faster decline. And people have found that seizures are associated with faster decline and more amyloid and tau pathology in the brain. So this is a really solid finding now that many people are following up on around the world. So back in the lab, we started studying different anti-seizure medications that could suppress epileptic activity in our Alzheimer's mice. And we found one drug called levetiracetam, or Keppra, that was very effective at stopping these epileptic spikes, as you can see at baseline compared to 2 hours after the administration of this drug. So based on these exciting findings, we began a phase 2 clinical trial funded by the Alzheimer's Association and by private donations and the NIH, where we screened people initially for cognitive testing and Alzheimer's biomarkers. That we did that overnight EEG and 1 hour magnetoencephalography just to distinguish those with and without epileptic activity in Alzheimer's disease. Then we randomized them into 2 groups. One group received placebo, followed by a break period, and then low doses of this anti-seizure drug, Lev. The other group received the same treatments in the opposite order, starting with Lev, followed by placebo. And then we did some cognitive testing and other assessments along the way. And the 2 groups were very well matched in terms of their age, and nearly 40% had epileptic activity. And there were 2 tests that were remarkably affected by the levetiracetam. One is this test called Stroop test. So in the Stroop test, we asked people to name colors just to make sure that they are able to see colors properly. So they would say yellow, green, red, blue, and so on. And then we have a test where we ask them to name the color of a word when the meaning of the word is incongruent with the color. So rather than blue, they would be asked to say green. And here, instead of yellow, they would be asked to say blue. And then rather than blue, yellow, and green, red, red, green, blue, yellow, green. It's kind of hard as you go along, but it requires a lot of mental flexibility, and performance in this test correlates with people's ability to problem solve and multitask in the real world. And what we found is that overall, the levetiracetam did not improve performance in people without epileptic activity in Alzheimer's disease. But in those who had detectable epileptic activity, this treatment improved their performance dramatically by 7 words in that 1-minute test. and that was a remarkable improvement. We also found that we tested how well people did in a maze where they were learning a route through a virtual town. And this is a test of spatial learning and memory. So we were able to test how they did on placebo versus on the drug. And if the slope trends upwards, that means that they're improving on the treatment. If it slopes downward, that means they're getting worse. So in the group that had detectable epileptic activity, these patients improved while they were on the drug, and they worsened when they were on placebo in their ability to learn this maze. So that's more evidence that this drug is helping just those patients with Alzheimer's disease and detectable epileptic activity. In the people without detectable epileptic activity, there was no difference between the treatment and the placebo. So we reported these findings in JAMA Neurology in 2021. So overall, I've shown you that this drug levetiracetam, which is now available in a generic form, is well tolerated. We know that it's quite inexpensive. And by comparison to this new amyloid lowering therapy, Leqembi, it's much less expensive. It did show disease symptomatic improvement in those with Alzheimer's and detectable epileptic activity. And we and others are beginning to look at whether it could also slow the disease course and improve outcomes in the long run. And there are several studies ongoing to do this. One of these studies at the bottom here recently reported their findings. These studies have been limited during the pandemic. There was evidence from this Hope for MCI study that the low doses that we used for LEV might actually help people who have certain genetic makeup, those with Alzheimer's or mild cognitive impairment and who are not carriers of the ApoE4 allele, showed strong trends towards improvement over a year and a half with this drug. However, they did not pre-screen for epileptic activity, and that is actually, we're seeing to be needs to be a crucial aspect of studies that investigate this medication. When I present this work to my clinician colleagues, I give them some clinical pearls, essentially about which drugs seem to work best in our older adults and Alzheimer's patients with epilepsy. And so levetiracetam in the clinic is actually quite well tolerated as long as people are not too advanced in their dementia. There's another related drug, brivaracetam, that has lower side effects, but it's more expensive, that we sometimes use. We also use a drug called lamictal or lamotrigine quite a bit in the clinic. And some of these other ones are not as good. And I make sure that my colleagues know that there are certain medications at the bottom of this list that can actually have too many cognitive side effects and are not good for our patients that are memory impaired or having other cognitive symptoms. And I recently wrote a commentary because there was a study by a group in France where they found that anti-seizure medications in patients with known seizures in Alzheimer's disease allowed for them to progress at a much slower rate than people that have previously been reported to have seizures or epileptic activity and Alzheimer's disease. They did not directly compare, so I was just speculating that potentially these anti-seizure medications could be slowing the course in these patients in this study that was done in France. And since our report of seizures that can precede dementia, many people have begun to think about whether new onset seizures in older adults that look fine otherwise could be a harbinger or a risk factor for a future risk of dementia. And groups that, including this study called ARIC, or atherosclerosis risk in the community study, and other studies like at the Veterans Hospital, as well as a study in China, has shown that late onset epilepsy, meaning epilepsy that begins generally in people older than age 60, increases the risk of Alzheimer's disease by threefold. And there's a risk factor for Alzheimer's disease, the ApoE4 gene, which about 25% of people of Caucasian descent carry, increases the risk of late onset epilepsy. Finally, people have shown that there are changes in the blood that are consistent with Alzheimer's disease that can predict the development of epilepsy, even in people without Alzheimer's disease. So there's a clear connection between late onset epilepsy and Alzheimer's disease in older adults. And one of the fellows in my research lab here came up with some guidelines for our clinician colleagues on how to assess people with new onset seizures of unknown origin. When seizures happen in older adults, oftentimes we find what causes that seizure. It could be from a stroke or a tumor or some other lesion in the brain. Sometimes it's also due to medications or metabolic imbalances. But up to half the time, we do not find the cause of the seizures. And when that's the case, we are proposing that in the clinic, that these patients undergo a thorough evaluation of their cognitive abilities, as well as testing for any evidence of Alzheimer's disease. And so the way that we clinically approach patients with late onset epilepsy is beginning to change based on some of our more recent discoveries. And we can divide people into those with late onset epilepsy without evidence for Alzheimer's disease, or with clear evidence of Alzheimer's disease, or with intermediate evidence of Alzheimer's disease. And so these patients will, I think, begin to get more attention in the clinic. And that's important because we know that these amyloid lowering therapies are most effective in the earliest stages of dementia. We also began to look at people that come into our hospital for surgery due to intractable epilepsy. That's epilepsy that is untreatable with current anti-seizure medications. And we studied the tissue from these patients that had temporal lobectomies. And many of these people were quite young, anywhere from their 30s up to their 60s. And in this series of 14 individuals, nearly half had evidence of amyloid deposition or tau neurofibrillary tangles in the sections that had been taken out of the brain, indicating that maybe the seizures are causing some Alzheimer's-like changes in these patients. Here are some examples that are quite striking based on the young ages of the patients. This is a 62-year-old woman who had epilepsy and hippocampal sclerosis, or scarring in the memory structure of the brain. And she had quite a bit of amyloid plaques throughout the section that had been taken out. This is a man who had neurofibrillary tangles in the hippocampus after having that section of the brain taken out due to epilepsy and scarring in that region as well. So the people in these ages should generally not have any signs of Alzheimer's disease, but potentially the epilepsy could be triggering some Alzheimer's-like changes. We're also looking at UCLA at genetic risk factors, and we have a study called Atlas, which tracks people according to their medical records and also collects genetic information on thousands of individuals. And with thousands of individuals, we're able to narrow down genetic regions that confer risk for epilepsy or Alzheimer's disease. And we also were able to find genes that were where there were variants in these genes that were associated with increased risk of epilepsy and Alzheimer's disease. They had a shared connection to both diseases. And some of these proteins, which the font's small, but one of them is BEN1, and this is a protein that I'm studying in the lab related to the CART-funded project. And so I will go into that here in a moment. We recently wrote a review article discussing all of the various mechanisms by which seizures we think are occurring in Alzheimer's disease. And this can occur both within neurons, and this is the end of a neuron called an axon, and this is a synapse, essentially, with one brain cell communicating with another through a neurotransmitter called glutamate. Well, glutamate excites cells, and in Alzheimer's disease, there's excess glutamate at the synaptic region that activates receptors like NMDA. And when this receptor gets activated, that leads to activation of the next cell. And when this occurs too much, that can lead to seizures. So we're looking at connections between tau and molecules like FIN and the BEN1 that I mentioned that can regulate neuronal activity. FIN, for instance, increases the responsiveness of this receptor to glutamate. And so we're looking at how tau is involved in this process. We're also looking at, and others are looking at the glial cells. These are the immune cells, which regulate, they can actually take up this glutamate, and they can also have other effects related to potassium currents that can regulate neuronal activity. So one reason that we're really interested in tau is that we can remove tau from our Alzheimer's models genetically. And when we do that, these mice have improved learning and memory, even when they're also expressing amyloid beta plaques. So here's an example of a Morris water maze where we have mice learn how to swim to a platform that's hidden under the water. And we do several tests over many days until they learn where that platform is, because they're very motivated to get out of that water. And then we remove the platform and have them look for that platform. And once they've learned where it is, they tend to swim around that quadrant of the pool looking for the platform. But in the Alzheimer's mouse, you can see that it's wandering all over the place. It doesn't remember where that platform was. But in this Alzheimer's mouse that has the amyloid plaques, but the tau protein has been removed, it's able to find that platform effectively, showing that the memory is retained. This was a seminal paper that many have followed up on, including myself. This was a paper that came out of the lab where I was just beginning my fellowship training. And in our models of Lewy body dementia, my colleagues in Minnesota found the same thing. They found that when you remove the tau, these mice learn how to escape this platform called a Barnes maze. They can find this escape hole much better than they could when they have normal amounts of tau. And related to the epileptic activity, we found that removing the tau is very effective at suppressing the epileptic activity. And that is true for both the Alzheimer's model as well as the Lewy body model. Here's a tracing of a Lewy body model that overexpresses alpha-synuclein with mutations that's having multiple epileptic spikes. But in the same model with the tau genetically removed, there's only one spike in this whole tracing. And we've also found, as I mentioned earlier, that the tau improves the learning and memory in both models when you remove it. So based on these findings, we're now looking at this cascade of events that's occurring, initiated by proteins like amyloid beta or alpha-synuclein, that leads to epileptic activity. The brain begins to remodel to prevent the epileptic activity, but that leads to cognitive decline. And we can prevent this cascade of events through multiple mechanisms. The tau mechanisms are mechanisms that I began studying with the initiation of the CART funding in 2018, which enabled me to get NIH funding through an R01 grant, as well as another grant that we recently got here at UCLA. And then we're also looking at anti-seizure drugs, both at a study here at UCLA and another one at UCLA focused on the Lewy body model. And we are making good progress on these two avenues. We've made great progress with anti-seizure drugs, as I mentioned earlier. So in the studies that we're now reporting, based on the work that we started with the CART funding, we have mutated this tau protein in a certain region called the proline-rich domain. And that's a region that allows tau to bind to some of these molecules that affect neuronal excitability, including BIN1, FIN that I mentioned earlier, PLC gamma, and P85 alpha. These are molecules, mostly enzymes, that regulate cellular pathways that can lead to hyperexcitability and seizures. Tau is a protein that has many domains that bind to other molecules as well. And we recently did a very thorough investigation of what these mutations in these specific sites do to its binding to all these molecules. And we found, interestingly, that we could prevent binding to certain proteins, but then that increased binding to other proteins. And we're looking at the functional effect of that as well. So our current studies in the in regards to these findings in the humans are to look for epilepsy in Alzheimer's patients using hippocampal recording and people who are suspected of having these silent seizures deeper in the brain when we're not able to detect them through scalp recordings. And so this is a study that I'm working on with my epilepsy colleagues, and we're going to treat our patients that have hippocampal seizures with an anti-seizure drug and see how well they do over a year. This would be the first of its kind type of study where we're actually connecting a wire to a brain region and recording directly from the brain in Alzheimer's patients. We're also beginning to look at other markers of seizures and hyperexcitability and using MEG and EEG to do that. We have an interesting study where we're looking at music stimulation to improve brain rhythms and improve cognition. And then I've been expanding my studies here at UCLA into underrepresented populations in order to begin to assess what we're seeing in groups that have not traditionally been studied as carefully. And in regards to the MEG, there are many brainwaves in the brain that range in frequency from 2 hertz up to 30 hertz or even more. And we can see how well those brain rhythms are in sync with each other through a test called global connectivity. And we've begun to look at that because we know that epileptic activity is one feature of disease, but there could be other features that are easier to detect, because in order to find epileptic activity, we often need to record for 24 hours. But there may be some other brain rhythm abnormalities that indicate that epilepsy is likely that could be detected even within 10 minutes. And one of those tests actually showed quite a good difference between those patients with Alzheimer's and epileptic activity and those without. And that is the alpha connectivity and the delta theta connectivity. The alpha rhythms are in the 8 to 12 hertz range, and we found that they dropped out in the back of the brain in those Alzheimer's patients with and without epileptic activity. But you can see the blue color here are the regions that are affected with alpha synchrony abnormalities. And the alpha synchrony abnormalities were more pronounced, you can see here, in those Alzheimer's patients with detectable epileptic activity compared to those without. And we also saw increased synchrony in this delta range between 2 and 8 hertz, delta and theta, that was much more pronounced in those with the epileptic activity. So we're beginning to think about this short 10-minute MEG exam as a way to distinguish our patients with and without epileptic activity, which is really critical in identifying patients that need anti-seizure medications. At UCLA, there are colleagues in the epilepsy department, including Anatol Bragan and Jerome Engel, who discovered that there are these high-frequency oscillations that indicate risk for seizures. And these are oscillations that occur in the much higher frequency ranges. And these are really extremely high frequencies that we usually don't even look for in humans. But we see many of these bursts of activities in our models of Alzheimer's disease, as well as our model here of Lewy body dementia. They're much higher in frequency than in our controls. So we're beginning to look in our human models, or human patients living with either MCI or Alzheimer's disease, and we're seeing quite a bit of these abnormalities throughout the brain. In these bar graphs, you can see that throughout all the different regions of the brain, there are more of these high-frequency oscillations in the patients compared to the controls. And in a work that we're going to publish probably this year, we found that treatment with that drug, levetiracetam, helps suppress these HFOs, but only in those patients who had epileptic activity. So that's a really interesting discovery that could help us think about HFOs and measuring them more in clinic. We also are doing the study of music stimulation. And what we're doing with music stimulation is that we're altering music, and our participants are able to pick a playlist of music that they enjoy, but we alter it just a little bit so that the lower frequency tones are enhanced. And we're really focusing on enhancing the 40 hertz rhythms, which we think are really important for many cognitive functions, and they are impaired in dementias. These are the rhythms that get enhanced when you're in deep meditation or when you're more creative. And we've been able to show that we can have people listen to this music that has adapted sounds, and they have improvements in their gamma rhythms in the regions that are engaged with the music. So this is great because everyone loves listening to music, and it can have dual benefits. Both they can enjoy the music that they're listening to, but also it can have long-term effects on their brain rhythms and ultimately maybe even their learning and memory and other functions. This study was picked up by our local news station, Fox 11, and they reported on a woman who flew down from Alaska with her husband just to test out this protocol. And she enjoyed it so much that she's now testing it out at home. And I had mentioned that we're expanding our studies throughout greater Los Angeles. And that's because we really want to gather an abundance of information. In addition to EEG, we're looking at where people live and their lived experiences. These are considered social determinants of health. and how that influences their brain health, as well as risk for dementia and epilepsy. We're looking at genetics and other factors. And centers beyond the UCLA Westwood, we're looking at Olive View Medical Center, which is in Sylmar. It has a large Hispanic and Latinx population. Harbor UCLA as and MLK Outpatient Center, which also see diverse populations. This is through an NIH-funded mechanism called a UH2 grant. We have a wonderful team that I work with here at UCLA, and just wanted to acknowledge what a large team we need of clinicians and clinical research coordinators. My right-hand person, Stephanie, who kept me on track today to make sure I didn't miss this meeting. And then our database team and people that work at all these different hospitals. So it's really a dream team. And the name of our study is called DREAM LA, which stands for Dementia Research, Education, and Advancement in Los Angeles. So far I've shown you essentially that the seizures that our patients are experiencing may be really the tip of the iceberg with regards to brain rhythm abnormalities, and that silent epileptic activity can occur in over 40% of our patients. Together, up to 60% of Alzheimer's patients may be experiencing epilepsy at some level, either the silent epileptic activity or more obvious seizures. And we're also studying brain rhythms during sleep because we know that the seizures can disrupt sleep, and poor sleep can exacerbate seizures. So we're looking at sleep as well as brain rhythm changes in our diverse populations here in Los Angeles. And ultimately, our goal is to block the effects of the abnormal brain activity on the brain's production of these disease proteins like amyloid beta, tau, alpha-synuclein, and because those in turn can lead to more abnormal brain activity. And so we want to stop this vicious cycle from occurring by detecting the changes early on, treating them through various means, both anti-seizure medications, as well as the tau-based mechanisms that I'm studying in the lab. All right, so with that, I'd like to thank our growing team here at UCLA. This is the center that I run, and many of the my esteemed colleagues that I showed you earlier. And I have many epilepsy colleagues that I work with, as well as I want to acknowledge people at UCSF that helped me get started in this area. I still fondly remember visiting you all back in 2018 and receiving the first large check that I've ever gotten. And that was very memorable and fun experience for me. So I want to thank you from the bottom of my heart. I couldn't have done any of this without the funding from the CARD Award. In fact, that first R award that I got from the NIH was made possible through CARD funding. And they explicitly expressed that they wanted to see that I had evidence of extramural funding for my concepts before they would fund the study in a larger way. So can't thank you enough, and really enjoy talking about this work, and very excited about the directions that we're headed. I'll just give you some brief responses to all of them so that I'll do it quickly. So in regards to dementia therapies that improve cognitive abilities, we've for the last 20 years had some drugs that help with symptoms of dementia. These are drugs like Aricept and Namenda, and they are actually quite effective. They can essentially stabilize symptoms for up to a year, but they do not slow the progression of the dementia. So when people are taken off the medication, the progression is the same as it would be, naturally. So that's kind of where we are with symptomatic treatments. What are your thoughts of whether the cause of the dementia may be epileptic in nature? That's a good question. I think we're thinking that potentially some dementias are initiated through epilepsy, and that there may be a genetic predisposition for dementia in some people with epilepsy. The next question is, why has there been talk about whether tau and amyloid plaques may be a consequence rather than a cause of dementia? We probably don't talk about that enough. And so I think in regards to the tau, when we see it accumulate, that could certainly be a byproduct of stress in the brain. And we actually don't know if those tau clumps that occur inside of cells are causing the cells to die. In fact, it may be that that's the cell's response in order to stay alive. And so we actually, in our lab, think that tau is involved more in the early stages of dementia before it clumps up by binding to these molecules that we're interested in. And we suspect that preventing those interactions could prevent this cell mechanisms that lead to cell death. So in regards to the amyloid plaques, they're worried that maybe so the plaques we know for sure that the plaques themselves, which occupy up to 1% of the cortex of the brain, are entangling these neurons, and that certainly is not good for the cells that are nearby. But the brain can function quite well even if it were to lose 1% of its brain cells. So it's not that the plaques themselves are the only cause. We think that there may be other forms of the amyloid floating around causing dysfunction. And epigenetics is an area that is growing in the field, and certainly exposure to environmental factors like pollution increases risk of dementia, and that can have epigenetic effects that can alter one's gene expression. Epigenetics essentially means not just the genetic makeup, but how the genes are expressed. And epigenetic changes can even be passed down from generation to generation. So ancestors who have experienced stress can actually pass along some epigenetic factors related to that stress to their offspring. So that's a growing area of investigation. We haven't really thought about we thought about it, but we haven't really pinned down any epigenetic causes of seizures in dementia.
We certainly appreciate your comments about the impetus that your CART Fund grant had on your ability to achieve larger grants from NIH. That's something that we are very proud of, that we're able to do to support researchers around the country, and we certainly appreciate that comment as well. Oh, Dave Brenner does have a quick question about music, and of course we partner with Music Men's Minds and several other organizations through our Alzheimer's and Dementia Rotary Action Group, and so we've heard quite a bit about music over the years. And Dave Brenner was asking, let me get back to it very quickly, if music can help. Do you think there are certain noise pollution that can cause issues as well?
Yeah, it's certainly possible, because we're really focusing the music on refining it in a way that can focus on those gamma rhythms. But people who live nearer to freeways, we think due to air pollution, have a higher risk of dementia. But I don't know that we've really considered the noise pollution related to that.
We thank you so much for joining us today. I remind you that, again, we have 4 days left in the end of this fiscal year. We are saying thank you to Rod Funderburk, who has served as our president of this board for the last 2 years and has done an incredible job, and will be moving into the immediate past president's role beginning next week. We have lots of new folks coming on board that we're excited about. We have all kinds of new things planned for you guys in the coming year. So please be sure to, if you have not already, sign up for our monthly newsletter. You have to opt into that. You can do that by going to cartfund.org, and in the upper right hand corner there's a box where you can subscribe to our newsletter that goes out monthly. And then also, of course, if you will register for these events in the DACDB calendar, then you'll get the notifications via email once you've registered, and again the day before the event, reminding you with the links and all of that. The next lunch a la carte again will be on Wednesday, July the 17th, with Dr. Bill Parker, it just left me. Dr. Lessering, Dr. Lessering, I believe it is. And then our next monthly conversations call will be on the very next day on Thursday, July the 18th, because that is going to be on the third Thursday of every month. And of course, our annual training session will be on Monday, July the 29th, also at 5:30. And we look forward to seeing you all then.